problems now.
Wait, what's our team name?
Team
Uh,
Blue. Team Blue, I think. That's what I've been like just recording
Team
as
Blue.
that.
Yoika, what the
Sorry.
heck? That name is lame.
Well,
yeah,
if it bothers you, I guess we could change.
Here, let's do the poster behind Natalie's head and call ourselves Team Harry Potter.
I'm cool with that.
Harry Potter, yeah, that's on that email. All right, I will post that to them and let them know. All right, well, so do we just go through them like we've done before, or?
Yeah, pretty much. I think so.
Yeah, I think so.
All
First
right.
though, are there any like specific questions that you guys have?
Actually, yeah, I do. I wanted to go over problems 5 and 6.
Okay, let me look at what those are.
So,
Yeah, so I
Um,
only had a question on 5C, but I emailed her and she explained it. So
oh yeah, if you could explain that'd be great because I was definitely a little bit confused there too.
okay,
check it out. Here's
should
my
we
homework problem.
go get to that or start with it?
What?
Are we gonna start there or should we?
Maybe let's start with the questions we have because that's the most important. And then
Okay.
if we have time, we can go through everything else.
I agree.
All right.
Okay.
So let's just go. Did you want to go through the whole question 5, Nick?
I do, yes. And question 6 as well.
All right.
Yeah, that'd be nice.
So. Okay, so I'll go ahead and read it, I guess.
Okay.
How many bar bodies will be found per cell in the following situations? So an X-sick is deleted from one of the 2 X chromosomes in an XX female. Okay. So I got 0.
What did I say? Okay, why did you say 0?
Okay. Hold on. Let me go ahead and pull up the reading too.
Okay, yeah. I actually have it right here. You guys can see my screen right here.
Yeah.
With the PowerPoint right here. So anyway, if you go to the part that talks about, yeah, here we go, the inactivation center right here. So
Mhm.
basically, if it has more than one inactivation center, those additional ones are those X chromosomes with the additional ones are canceled out, right?
Mhm.
Okay, so from this one, it says it's deleted from one of the 2 X chromosomes in the XX female. So wouldn't that mean that there's one bar body?
Yeah, that was sort of what I was thinking too. Like I had to
Well,
go through Google and try and figure that out, but it still wasn't totally clear to me about why
okay.
there was still one. Or there wasn't one. I mean, there's 0.
So
Oh, okay, wait. It's again. Sorry.
So here it says X inactivation is controlled by a region of the X chromosome near the centromere called the X inactivation center (XIC). If the XIC is missing from one X chromosome and is present on the other X chromosome, then both X chromosomes remain active. 2 X6 must be present for one X chromosome to be
So
activated.
that would create a lot of problems, though, wouldn't it? If you didn't have, if they both remained active, that's just too much, right?
Yeah,
So that would end up being an issue.
probably. Yeah.
Okay, just
Uh,
wondering.
I do not know what sort of issue though, but
Yeah.
this.
Okay, so that makes a lot
Yeah.
of sense now.
Okay, so do we go on to the next one, or are we clear there?
Well, what do we decide that what the answer was then?
0.
It's 0.
It's 0.
Yeah.
Yeah.
Someone else want to read the next?
Okay, so are we doing the one, an XX female in which both copies of the T6 gene are non-functional? Assume that both copies of the Xist gene function normally.
Mm-hmm.
I think the answer to that one was 2 bar bodies.
That's what I said too.
I said,
And
What'd you say, Joachim?
I said 0. So, let's see, the T6 functions normally. Let me see. And
well, I think it's the T6 doesn't function normally, but the Xist does function normally.
oh, are you on the third one?
No, we're on the second one.
We're in the second, yeah.
So
the
that one
Xist
is Xist is non-functional and T6 is functional.
Yeah.
Right. So if you look
Or
on my
wait.
screen right here, it says here that the Xist gene is the one that inactivates the and the TIX TISC, is that it?
T6?
6? I'm not sure.
Yeah, T6, let's call it that. Is the one that produces the RNA that's complementary to the Xist to destroy it.
Yeah. So the Xist is the thing that goes out, sends out some sort of RNA, right? And the T6 is functioning normally. Yeah, so if the Xist doesn't send out the RNA, it's not going to, because the T6 also sends out the RNA, and when they complete each other, those RNAs are degraded, so it's kind of almost like a protection for that chromosome to from that the T6 send out, so that it remains active. So if the Xist is non-functional, it's not going to send out those RNAs at all, so it's not going to degrade anyone. Or
Right,
nothing
so
is going to
therefore
get degraded,
the
and
X
we're not going to create a bar body.
Yeah. Okay, yeah, I think I was looking at 3 when I was saying 2, so now I'm
okay.
understanding. I was like, where are you going?
Okay.
Just
Cool.
read the wrong thing. Yeah. I understand that one. And then, does someone want to read the next one?
Nick, go ahead.
Sure. An XX female in which both copies of the T6 gene are non-functional. Assume that both copies of the Xist gene function normally.
Yeah.
Okay.
So that would be 2 since T6 isn't working, right?
Right. So what happens is that Xist sends out the RNA, and since T6 does not have anything to complement it and degrade it, both of the chromosomes are going to be targeted and become bar bodies.
So bar bodies form when they're inactivated or activated?
I think it's inactivated, right?
Mm-hmm.
Is it in? Okay.
So if those are both inactivated, I don't think you'll survive, because that means you're not going to have anything really coding, and you'll die. So
sounds reasonable to me.
Yeah. I mean, the only thing it really says on something. It's it contains a bar body is just a highly condensed X chromosome. So I just assumed that the X chromosome was activated, but.
Yeah, but it forms 2 though. But it doesn't sound like a condition that would be very stable.
Okay. So
yeah,
I guess then the inactivation then When it's inactivated, then it forms bar bodies, huh?
Yes.
Mhm.
Okay. Makes sense. You guys
Yeah.
want to go on to the next one?
Sure.
Go ahead.
I'm just seeing if there's anything that I didn't really understand. I think the phenotype stuff was pretty straightforward.
Are we gonna do D as well on 5?
Oh, sure. I keep forgetting that we have those.
Sorry,
Okay.
no.
So I can read it.
You want to
Okay.
read D?
Go ahead.
I can read it, yeah. So it's an extra copy of the exec is inserted into one copy of chromosome 21. Assume that both of the X chromosomes contain normal execs. Okay. Yeah, so that would be 2 again.
Yep.
Because you have an extra one, and that's going to inactivate another one.
Mm-hmm.
Okay.
Yeah. So 1 remains active. That's what they want.
So we're all good on that?
Yep, I think so.
Okay. Is there anything else that you guys in particular struggled with?
I wanted to ask about 6 too.
6. Okay. Where? Okay. Do you want to read that?
Sure. It says, what are the phenotype ratios produced by the following crosses? And, I'll just say negative, negative, I mean, I'm sure you guys can say that it's IGF2, but I'll just say, negative, negative for simplicity's sake. So the female has a negative, negative, and the male has a positive, negative. So if you cross those two, what are the phenotypes? For the first one, I said 50% would be dwarf and 50% would be normal. Is that what you guys got?
Yes. I think so. Let me double check. 6. Where is that? Yeah.
Is that what you got too, Joachim?
Yep.
Okay. So, yeah, I just wanted to be sure to make sure that I understood it correctly. So
the mother's genes are silenced.
Okay, that's what I was thinking. So the mother's genes are silenced. So the only ones that really matter are the males, okay? And since IGF2 is, I guess, dominant in this case, like half of them will have IGF2 from dad, and half of them will have IGF2 negative from dad, right? So that's why it's 50-50, right?
Yep.
Mm-hmm.
Okay. So, and for B, kind of the same thing, except I said 100% of them are going to be normal.
Yep. Same here.
Yeah, that's what I said.
Okay. Now, for parts C and D, it says like a bald female and a heterozygous bald male.
Yeah.
So that's like referring to not the IGF2 gene, though, right?
That's the sex influenced
inheritance. I wasn't sure if it was the IGF. I don't think it was, but I mean, we know that, like, if a male has the gene for it at all, he'll be bald, and so I just used, like, what we know about the phenotype to figure out how to cross them. That makes sense?
Yeah. On my screen right here, if you guys can see it, I thought it was referring to the sex-influenced inheritance right here.
It is.
It is? Okay.
Yeah. So,
did you have a question on it?
Nope, I think I understand it.
No, I'm good on that one. Although I did, I don't know if you guys had a hard time, but, like, at the beginning when it was asking for definitions of, like, sex influence versus sex linked, I had a really hard time distinguishing between the two.
Mm-hmm.
I kind of got it, but I'm still a little shaky on that. So.
Okay.
Okay, so like a sex limited one
Mm-hmm.
is when they're like present on autosomes. So like and it is like determined by hormones. So like different genders will express different things.
Yeah.
Sex influenced is when the dominance pattern is kind of reversed often, as in like the baldness.
Male pattern baldness, yeah. Because just for a male, if you have it, that is dominant. Because you don't have anything to counteract it.
Right. The most important thing for the sex influenced is to remember that don't like focus on the letters too much, because usually like the capital letter is dominant and the lowercase is like recessive, like it's true for one, that still holds true for the other, but like the other, it's different, like for the baldness, like we talked about, like capital B and lowercase b is bald for guys, but capital B, lowercase b is not bald for girls. Does that make sense?
Mm-hmm.
Okay, yeah, that makes sense.
It's also found on the autosomes, or autosomal chromosomes.
Right. Or is that the sex limited Gokum that's found on the autosomes?
Both are.
Both are? Okay.
Mm-hmm.
Okay, and then like each gender produces like whatever hormones to initiate those genes, right?
Yeah.
Okay.
I know with male pattern baldness, that's also linked with like dihydrotestosterone, which I don't have. I have testosterone, but I don't have dihydro, and that's what like kind of will convert that. So, yeah. Okay. Did you guys have any other questions?
So for X-linked inheritance, just so we're clear, that one is basically just like when the the male only has one X chromosome, and it's like X-linked inheritance and stuff like that.
Can only get it from mom.
Yeah, can only get it from mom.
Good.
And so I think that's what it's referring to. It's like you can only inherit it from either your mom or your dad, but not both.
Yeah, that's what I was thinking too. Okay, cool. I just wanted to make sure on that.
Yeah, that's good.
Yep. I don't have any questions.
Yeah, I think we covered pretty much everything, and the questions we didn't cover are the same concepts that we already did.
Okay.
Yeah. So I mean, she did say, like you said, that we're gonna go over the things that we have problems, like questions or problems with. So, and I think we've covered that and so on. So.
Okay. And did you guys want to plan for next week now?
Sure.
I could do this time again, or if you wanted to, like, because you said the morning tomorrow, like, because I can do Friday mornings also.
So the thing is, like, I'm from Sweden, and I'm off track, and I'm home right now, so I'm 8 hours ahead of you guys.
Okay. So if you tell us times, I can probably work better, and it's probably easier for you to say.
Thursdays, like I've had 2 other Google Hangouts today already, and we've like, kind of set that as the day. So like I don't mind doing all of them on Thursdays to like get them all out of the way.
Yeah, Friday morning's tough for me because I have class at that time.
Oh, okay. I can do this time again if it works for you guys.
Is 2 the earliest you can do?
Usually I can probably do before 2. Today I just had a lab report that was not done yet. So I could do like 1. Or no, wait, yeah. Thursday, yeah, I could do 1 on Thursdays.
Okay. So is that okay with you, Nick?
Yep, 1's fine.
Okay.
All right. So
that's like 9 p.m. for me, so that should be good. If anything comes up, I guess we'll just have to email.
Yeah, that works.
So yeah, let's plan on that, and I will go ahead and tell them that we changed the names. Oh, by the way, I'll probably stop recording right now.
Okay.